All right, YouTube, let's talk about this stuff. Biotech, eugenics. We're going to do a review of where things are here in 2021 and where we need to go. Naturally, that is up here. So, most people are familiar with this movie, Gattaca. Came out in late 90s or something like that. And it describes this kind of extremely divided society where there's the genetically modified people and the kind of naturals, and the naturals are quite inferior. And then they implement some like very irrational discrimination based on this. Instead of looking at how great people are, they only look at the genetics. And no, of course, no sensible person would do this, but it creates a story. The interesting thing about this movie, aside from it always being used by people who are stupid, is that there's a scene in the movie where a couple, that's the father and the mother, parents of the protagonist and his brother, they go into this clinic as an embryo selection clinic. They go in, they look at the various embryos they can choose from, and the doctor tells them about the various trade-offs, or he even suggests some genetic editing or splicing in some genes from a different person to get extra sporting skills or something like this. And that is quite prescient from the perspective of a late 90s movie on this topic. In fact, to promote the video, they went even further. They make this amazing poster, this troll poster for the movie to really stir up some trouble, and where you could a child you can mate to order, and how far will you go, and you can look at all these traits that you can select for or against: criminality, gender, myopia, obesity, athletic prowess, and all these things. Of course, intellect is one of them. So of course, here we are 20 years later, and reality is quite close to doing these things, or even there in some points, to some degree. So in this video, we'll go over the eugenics, and that is the improvement of human stock, or the improvement of human genetics, and improvement in the sense that we take it from whatever the current stage is, we make it so that the genetics produces more traits or better traits in line with our values. And we'll go over first classical eugenics, which is the one that most people bitch about, and the various forms of it. Generally speaking, you can break it into two forms: the positive and the negative. The positive is essentially about promoting births among superior people, by whatever definition of superior. And negative is about decreasing birth among inferior people. And so these might be criminals, or they might be, in case of superior, might be healthy people, or in inferior could be people with epilepsy or something like this. There are many ways to do this sort of thing. You can have school curriculum that promote or try to convince people who think they have bad genes from not reproducing, or you can have the media saying that good people need to produce more. You can change taxation rules so that higher earners or smarter people or more educated people, or whatever proxy you want to use, they're exempt from tax or something. And of course, the most extreme method is you can essentially you can force people by sterilizing people with the state believes to have inferior genes. Anyway, that's the standard old stuff. Modern biotech eugenics is quite different. It's very liberal in the sense of being about choice and freedom, and that's why it's also called liberal eugenics. And of course, also to make the lefties not hate it so much. As we'll see, this is a bit ironic. There are basically two forms of modern biotech eugenics. You can either choose between siblings and embryo selection, or between gametes, as we'll see, or you can directly try to edit things, or even in case of editing, you can also move genes from a different species if you're this brave, or maybe from Neanderthals, who knows, or between races. Anyway, classical eugenics, the normal view, and of course here we want to say that we should be a bit beware here, because we know that politics biases everything, and it's not possible for humans to leave their policies back home, so to say. It's just not humanly possible. And we know in history from this study of registered voters that there's roughly 18 Democrat voters per one Republican voter among American historians. This value may be a bit higher because some people who vote Republican, they don't register, and you can't see them, they're hiding it. So maybe it's only 15 to 1, or maybe even 12 or something. But it is some very large proportion that is Democrat or left-leaning. And the typical history of eugenics and how it was used. You can get this in the name of eugenics. As you can see, it has on the book cover. Stephen Jay Gould says it's awesome, so it probably sucks, right? So let's go over this sorted history. Essentially, after Galton, he came up with the eugenics idea. Nothing much happened for a few decades, and then some people decided that we need to start making records so we can see who's breeding, and so they make these pedigrees, right? So family trees. You can see who's descended from whom, and it was obvious from this that some families tend to produce tons of misfits, and because crime and misfits and social benefits and this sort of thing, they're very expensive to society. The society has a strong interest in reducing the frequency of such people. And so, for instance, I found some statistics here. North Carolina, you can see they sterilized a lot of people. The interesting thing to note here is that World War II ends in 45, and you can see actually most of the sterilizations are after 45. So it wasn't just that things immediately stopped after World War II. It was only in the 60s or so things really finally stopped, and that really, of course, has to do much with the cultural revolution of the 1960s, and maybe not so much with this World War II business that we're told about. As with everything good, there is a German who will take it too far, and in Germany here we had the Extreme Action T4 program, and I even found this. So according to Wikipedia, some 300,000 Germans or so were killed, and this is an involuntary euthanasia program where essentially the state monitors who has deficient kids, whether they're retarded or epileptic or Down syndrome or some other obvious likely genetic problem. The state essentially went in and said, look, we don't have enough resources to support these people, we should just kill them. And when you try to kill people in a nice way, we call it, of course, euthanasia, good death. And this was mandatory, and a lot of it was psychiatric, and apparently it's done by the SS. So that's not too nice. And of course, everybody wants to talk about the Holocaust, and you can see why this makes sense if you think that problems among people are caused by their genetics, and you cannot fix things with social programs. It's kind of inherent to people. Then you can try to make some people have fewer kids, more kids, and maybe not be too dramatic about it. But if you really think that an entire population has nothing of value to add to humanity, I mean, the obvious answer is to get rid of it. And unfortunately, in Germany, there were some people who took this very, very far, and now we always hear about this. The thing they don't tell you about this classical eugenics is that despite most people being against it nowadays, the people are leftists. The people who actually promoted and implemented eugenics in the early 1900s, these were actually also the leftists. Very interesting. It was actually the Social Democrats, and I found this picture of this Danish guy, K.K. Steinke, somehow has a German name, fitting, right? He was the Minister of Justice, and he implemented some of these rules. And it was true both in Denmark, Norway, Sweden, and maybe Finland, I think. Finland. They all implemented this social democratic eugenics as such, and it's essentially because they reasoned quite correctly that if you implement a welfare state, and the welfare state has to pay for people who are unable to take care of themselves, and such people multiply in your society because of the welfare state, this will sooner or later lead to a collapse. And so they were in fact thinking of kind of negative dysgenics or internal dysgenics, to use Helmut Nyborg's term, and not immigration, which is a bigger problem nowadays, and probably always was. Anyway, but the problem is, of course, that they don't really tell us there was actually left-wing people who implemented this stuff. They kind of pretend that it was all evil right-wing people. The Nazis came up with this stuff, and that is quite not true. History usually is a bit less clean than ideologues would have you believe. Anyway, after World War II, after World War II somehow, I don't know, and there was a decline in eugenics, as we'll see. But I mean, we already saw it. It mostly kind of fizzled out in more like the 60s or the 50s, not really immediately in the late 40s. And there was this overreaction by academics and various liars, such as Gould, Rose, and Kamin. And you can see why you would be this kind of, why you would want to distort things in that direction. These were all Jewish academics, and roughly half the European Jews died in the Holocaust. So if someone, I mean, think about it, if someone just had a big war and they exterminated half of your population, maybe you would be a bit inclined also emotionally to not be too much in fan of things that go in that direction again. Even if things, of course, also could help you. In Israel, there's a long history of eugenics, of course. In Israel, they're not very afraid of Nazis taking over and doing another Holocaust, so they're not afraid of eugenics. It's only normal Europeans are being shamed with this. This denial of the ability to change genetics, even for the better, is you still see it. Where Richard Dawkins, this year or last year also, got in trouble for saying that, of course, eugenics would work. Selective breeding works, of course. You can breed cows and whatever it is you want to breed, more or less. We have all these weird dogs now. They all come from wolves, and Chihuahuas don't look a lot like wolves. So, the really we need thing to do is that we need to decouple a bit the mistakes of the past. There should not really be a limit on what we can do today. A bad start of some idea or technology, that doesn't really mean that we should just never pursue it again. That's an irrational mindset. And for anyone who thinks about this even for 5 minutes, you can think of many technologies that got a bad start or had a bad, like, say, medium, but now are looking very plausible, or is something that we obviously want to pursue. Airplanes used to be terrible and killed a lot of people. Hot air balloons, we had the Hindenburg disaster and a lot of other disasters. Supersonic air travel, there are various military planes, but in terms of passenger planes, we have, of course, had the Concorde, which flew perfectly for a couple of decades, and then there was like one crash, and now they're like, no, let's get rid of it. And there are a bunch of companies who want to reintroduce supersonic travel because flying around continents just takes too long. We need something faster. Space travel, the Apollo program in America, when they want to go to the moon, the first Apollo mission, so to say, there was an accident, and all the initial team, they all died in a fire accident. But that really didn't stop America from going to the moon. They just made a new project and made some fixes. Ocean liners, there's tons of sinkings. The most famous one is Titanic. Bridges, there's the famous wobbling Tacoma Narrows Bridge. Dams, there's just too many dams that broke and killed hundreds of thousands of people to go over. Medicine is like the winner of bad ideas. Bloodletting, lobotomies, radioactive baths, alcohol for babies, methamphetamine for weight loss, and there is an endless number of these. Anyway, you get the idea. So modern eugenics, really you can check it by following in Google Ngram, that is the frequency of a word in books over time. And so we can see down here in 1883, Galton, he coined the term. I don't know why it exists slightly before. It should only be in 83, probably misidentified book. It's essentially nothing much happened the first like 20 years or so until we get to 1910 or 1905 or so. Then it's kind of skyrocketed. This was the first heyday, the initial peak, we can say. Then World War I happened, and there was a big decline, and then there was a slight resurgence after that in the 1920s, and then in 1925 or so we see the big decline starts. And this of course it does not really fit well with the usual narrative, in that eugenics was defeated by the Nazi abuse, and everybody afterwards realized, whoa, this was not really a good idea. As we can see, this is not the case. It started in 1925, and it kind of fell steady during World War II, and after World War II there was not a big difference. It was almost at the 1960s low, and then it lay dormant for another, slightly increased for the next 25 years or so, until we really begin the IVF saga era. So that's where we are from here and forward, roughly from 1980 or so, 1978 and forward. And so after this, the first few years, there were very few babies with this, but then in the 1990s or so, this technology really takes speed down here, and people start talking about eugenics again, because now we actually have a much better method than the sterilization method, which was or euthanasia, which we're cruel and not very good back then because science just wasn't ready essentially. So IVF really works kind of like this. IVF, in vitro fertilization or in glass fusion of the egg and sperm cells. And if you go to a typical IVF clinic, you get this process. You go for a consultation. Essentially, they're gonna tell you about what the steps that women have to go through, mostly women, right? Because you have to do some ovarian stimulations, not very nice. Blood work, initial, just to test ovarian stimulation. Essentially, you take women and you give them a syringe, and they inject themselves. And a friend of mine who went through this, she actually took a picture, a selfie of her stomach injecting herself. And what it does is that the ovarian stimulation, it makes women release more eggs than they would normally do. And so you know already that you can have twins, and some 2/3 of twins are due to women releasing 2 eggs at a time instead of the normal 1 egg. And what you do with this ovarian stimulation is that you stimulate the ovaries to reduce a lot of eggs. And then these eggs then had to mature, and you retrieve them. And so you have to do a kind of scraping. You have women lie down, and you literally scrape them out into a petri dish. Once you have the embryos in a petri dish, you're going to put a bunch of sperm from the man or the donor, whoever that is. Sometimes you use this method where you take individual sperm and you literally put them inside the egg. And this is used when the sperm quality is the problem. Like the sperm are not motile enough; they don't really move properly, and so it's a so-called paternal problem with the paternal infertility. Once you have a bunch of fertilized eggs, you grow them for some days, usually about 6 days or so. At this stage, they will have like 200 or so. The ones that make it, not all of them make it, as we'll see. And here's where you take the selection, and so you transfer, say, 1 or maybe 2 embryos. Usually, the way this was done in the past is that clinicians, they stand and they look in a microscope for the embryos, and they look at ones that seem to be growing well, and the ones that are growing well have a better chance of implementing. And implementing means that you put it back in the woman, and it has to get stuck there and start growing. But often what happens is that you put it back, and it doesn't get stuck for whatever reason. It doesn't implant, and it just fizzles out, and you have to wait until the next period where the woman is then fertile again. Anyway, so here's what you do with selection: is you take some tissue sample of these embryos you have that are grown to day 6 or so, and that small tissue sample has maybe 5-7 cells in it, and that's enough that you can sequence it or microarray, and that's when you can use the polygenic predictions, or you can also do the other stuff when just look at whether the chromosomes are the normal count. Then you wait to see if it has taken, and you can do a pregnancy test. And essentially, you redo this every time it doesn't work. This step only here, you only need this to get the embryo. So once you've done these first few steps, you have a bunch of embryos, and you can implant them one at a time. And if one of them works, obviously you have to wait until pregnancy is done and so on if you want to do it again. But that's the basic human the human cycle. In embryo selection in animals is already totally standard stuff. You can find many cow papers that have nice graphics like this, because with cows, no one cares about the past of eugenics and sterilization and stuff. They just want to make more money. And you want to make more money, you want to be very clear in your communication, and you don't want to have like a lot of the moral bullshitting. So we see much the same thing. We have a bull that's high quality, we have some cows, we collect the semen and the eggs. We do the same thing as with humans. Then we have a bunch of embryos. We wait until they grow, we take some samples out of them. We maybe do some editing, or we select between the embryos based on a genetic sample. And now in animals, they go a bit further. What they do here is that they also get a bunch of eggs from other cows, and then you get rid of the DNA in the egg, and you remove the one you had from the other embryo there. So you're just using the main part of the cell, and you're just swapping the DNA. And this has also been done in humans, and that's where you get the story with the free parent baby. Because the reason it's free parents is because in the cell here you have some DNA in the mitochondria, very small proportion, maybe 0.1% of the genome is a mitochondria, and which comes from a donor mother. So when you do this thing, this cow will contribute, say, 0.1% of the DNA, and the 99.9 from these two up here. Then you put these ones over here, indented, and you wait for them to grow, and then you restart the cycle. So that's how you do it in animals, and this is totally normal business. It's been done for a couple of years, or maybe decades or two. So we can see here, I found some statistics. Unfortunately, I couldn't find newer ones, but what you see here is the bovine SNP array. It's an array, microarray you use to do this measurement here, and you can see how many cows have been sequenced for this sort of thing, and you can see the blue line shows the improvement of milk quality. And so the green one is when they really started this, so 2008, that's when they started. So roughly they've been going for 15 years or a bit less, 13 years or so with this particular chip, and there were probably some before that. Citation is just like scientific index. So if we had this chart for new, it would probably be up here, right, somewhere. So that'd be nice. So I'm just saying that the business is moving. Anyway, so effectiveness of embryo selection is really quite complicated because it depends on a number of factors. You have first and foremost more eggs. That's just what you need. The problem is you do the scraping from a woman and you get a set number of eggs, and you essentially have infinite semen cells since they're just produced all the time. And so the eggs really is the limiting factor, the most important one. So what you really just need is more eggs. Women are born with all their eggs and they decay over time. So the eggs that women have, they're actually in the fetus when they were in their mother. Number of semen, spermatozoa, we covered this. They generate constantly. Paternal age effects, though much discussed, I think they're too small to care about, so I wouldn't really worry about this. The next problem is that when you try to fertilize the eggs with the semen, this doesn't always work. Even when you put this semen cell inside the egg, sometimes they fail to fuse properly. There's some rate here that you could optimize. The rate of embryo growth. Embryos, they grow the first 6 days or so, as I said, before you implant them, because you can't really do a biopsy of the very small embryos because you kill it by doing a biopsy. Obviously, if there's only 4 cells and you sequence 4 cells, there's no embryo. So you have to wait until there's enough of them, and we've just empirically determined that roughly day 6 or 5, that's when there's roughly 200 cells, and you can take a small biopsy with as the smallest cut you can try to do, and you can get these cells out. After this, but actually what happens is that many embryos, even when they're fertilized, they just fail to grow properly to this day 6 stage. Like they die on the way, or they grow but they don't get large. And in studies where you try to implement some of these not fully grown embryos, they sometimes work, but they work a lot less than the fully grown ones. And so there's some room to grow in improving this process, maybe some chemical stimulation. After this, you have the successful transfer, and so that's, you take the embryo that you wanted, you have done the test, and this is the one you want, you just implant it in the woman, but somehow it doesn't stick, it doesn't start growing. There's some successful chance here. I don't think we've been making much progress there. Then comes the things that we discuss a lot. It's the predictiveness of genetic models, and it's not just the between family, because all the embryos are siblings, and that is also true for gametes. And so you need to be able to not overfit to family factors, because if you overfit to those, it won't work between siblings. So ideally, you want to do GWAS on sibling contrasts or parent-children. So some kind of family control is the easiest way to deal with this overfitting problem. You can also maybe emulate this with appropriate statistical controls, but as you'll see later, we've been doing this for a decade or so, and it's not quite working correctly entirely for some social traits, which is unfortunate because these are the most important ones. Of course, heritability of the thing you want to select for. When you're doing embryo selection, you're really only looking at genetic variation. If genetic variation is not important for whatever thing you want to select for, then it's not working very well, right? It's fundamentally limited by the genetic variation. And to give an example, for instance, type 2 diabetes and lifespan, longevity, how long you live, these traits are not that heritable, maybe 20% heritable or something like this. So even if you had a perfect genetic model, you would only be able to you only have these 20% of the variants to deal with. The same is true for some other traits like homosexuality and many types of cancer. You really cannot do a lot of genetic selection here because they're just fundamentally not highly heritable traits. Finally, you have the unwanted genetic correlations to other traits. This is the scare one. We'll go into it more later. To go over the first one, egg harvesting. Here I found some numbers, and what you see is that you do the stimulation. That, remember, that's this one. We do this one, and we check. We get see how many embryos we get, or the eggs we get, and this is how many you get as a function of age. You can see that it declines, and so essentially you want to harvest eggs once you're in your 30s or maybe even 20s, and then you can save them in the freezer for a long later time. That's another thing I forgot to go over here, is that often you freeze them down and not all of them make it out of the frozen. Maybe you lose 10% or so. Anyway, so you want to do eggs earlier, but you can see you get like 15. The nice bonus here is that the more times you harvest eggs, the more you get. There's a kind of weird positive feedback. So if the woman is so inclined, they can undergo this the eggs harvesting multiple times, and then you would get maybe 50 eggs or something. So that would be very nice if you want to do selection. If you're already like in the 40s and you're getting like 7 eggs at a time, and the eggs you're getting are quite low quality, like maybe half of them would actually not work, since they won't grow, and they have multiple genetic major problems such as incorrect number of chromosomes. You don't have much to do with. You're just gonna have to harvest eggs tons of times. Unfortunately, you can only harvest once per month or once per cycle, and it takes 28 days. So that is you have a big problem there. Here we can look at the problem. Here is that the egg pipeline is quite leaky. So let's take this one value here that's from some clinic. They estimated some values. You start with 15 of these follicles. These are the kind of pre-eggs. They're not quite ready, and you stimulate them. So they say that, okay, 25% of the ones you stimulate, they just don't develop properly into real eggs, and then you have to retrieve some of them. Again, you have some problem. You didn't retrieve all of them because you kind of have to catch them in the uterus or in the ovary. Then you have to fertilize the eggs with the semen. Again, some of them will not get fertilized properly, even when you stick the semen into it. Then you have the growing the embryos, and you can see you lose another every time, and some of them are genetically abnormal, and these will be the ones that have, say, either Down syndrome or some other thing. Down syndrome is very famous because, well, it's viable, so you see babies born with this, but as a matter of fact, you can have incorrect numbers of chromosomes for every chromosome. It's just that essentially all of these other combinations of having, for instance, 3 copies of chromosome 1, it's not viable. No baby will be born with this. The fetus will always fail if you implement it, so there's no point in implementing it. Then you do the transfer, which may or may not, if it implants or not, and eventually you have to do the pregnancy, and of course all the women, they're going to fail often at pregnancies too. They're going to have a miscarriage, and that is the way life is. Humans have very high miscarriage rates, and eventually you end up with 1 baby. So 15 pre-eggs, 1 baby. There's a very leaky pipeline. There's a lot of room. You need to make these steps have less loss, right? They need to leak less eggs, or we just need a lot more eggs. We come back to the same conclusion: more eggs. Predictive models, as we mentioned, the problem is that when you do a GWAS, you want to control for genetic stratification, and some people who live different places, they have slightly different genetics, and they may be taller for some other reason than genetics, maybe kind of food they eat or whatever, and you want to control for this. And so the normal thing to do is include a bunch of principal components in the GWAS, and you say that we probably control for things. And there was a, I think, 2009 paper that showed that this works a lot better than not doing it. So everybody does it. Unfortunately, for some traits, you can see this plot is hard to read, but you may pause and figure it out. What you see is that once you do between families, that's the dark blue here, and inside families, there is a loss of validity. So the model is fitting to something that isn't exactly IQ. It's fitting to parental education that affects child IQ. Many of the samples are children, or they didn't actually use IQ. They used educational attainment, which is much more probably affected by parents, especially if it's like college or not college. Like college educated parents will push their kids to go to college, even if they have like doubtful genetic or intelligence to do this. And this overfitting creates this problem. There can also be genetic nurture stuff and weird other things that are getting complicated. Anyway, the easy solution, we train on sibling data. So what we need, we need millions of siblings that need to be genotyped. We are making such progress. Here's a newer study that's currently preprint, and here we see again the between within. We can see, for instance, see here for depression, there's a big problem. That's the between validity, I think, and this is one of the within ones. So basically, the model they have right now, it predicts nothing insofar as we can tell, but the error bars are massive, right? So clearly we need to do better. In terms of height, for instance, there's no bias, right? The height predictor is equally good within and between families, and that's the case. I don't know why BMI is fucked up here. Education seems to be working okay here, and so on. Anyway, so we need to go further though. Fortunately, genotyping is getting more and more common. Lots of people are paying for their own genotyping from these ancestry services. There's this one paper by Rashid Khan that has this plot. I couldn't find a newer one, but I'm gonna guess it's gonna be up here, right? A lot of people are bored at Corona and they're gonna get genetics genotyped. The problem with all this is that these are all microarrays, and microarrays are limited, as we'll see, but fortunately the price of sequencing has also been falling, and it's in fact falling faster than Moore's law. And especially in 2007 here there was I think the next generation sequencing or something was introduced, and so we're getting down here. We need to really go a bit further, because if you want to sequence tons of embryos, the sequencing shouldn't cost like 1,000 euro per embryo, right? They should cost 10 euros or something like this. And if you want to sequence tons of sperm, as we'll see, it needs to be much cheaper, right? Genetic engineering will cover this. We really have the CRISPR guy here, who's a Young Kui maybe, and he did some simple editing of embryos, and they seem to be born to healthy babies, and then he got suspended or even imprisoned. Anyway, so that didn't really go too well. China, however, is still making progress, even if they imprison their pioneers. So China has several projects currently where they're collecting DNA from millions of men. In fact, they're doing it from the army. They say that they want to do 10% of the male population in China. In China, remember, 1.4 billion people, half are male. Well, sort of half are male. Let's say it's 800 million male. They want to do 10% of those, 80 million males. That's a lot of men, right? If they sequenced all of that, they would be much larger than all European samples combined. And if they also have like military conscript rate, conscript data for these tests data, height, BMI, maybe a medical history, China is getting far ahead here. That shouldn't really be happening, right? But somehow we're sleeping on duty. The problem with genetic engineering, to go back to engineering now, China, is that when you do a GWAS study and you find, for instance, these 4 SNPs, they're associated, they predict, say, height, but that doesn't mean that they cause height differences. So the problem here is that if you change, for instance, SNP A, maybe SNP A is actually not important. Maybe it's SNP C or this one. So in this case, SNP A is the real one, and the reason these also show an association is that these SNPs are close on a genome to this one, to the first one, the red one. And so these also predict height, but changing these wouldn't do anything. They are just correlated with it. And so when you're doing genetic engineering, you need to change, say, a specific letter. You need to know which one is the one that actually does stuff and which ones don't do stuff. And currently we just don't know this. We have some ideas of maybe in total GWAS we find I don't know, 2,000 SNPs for intelligence, and maybe 10 of them we're fairly certain that these are causal. Okay, so we have 10 we can change now. Supposing we had good editing, maybe we could add 2 IQ or something. If you spent tens of thousands of dollars on this, it's not been done yet, and it remains to be seen. That's really the problem with this. So these are the tag variants because they just tag this one. The human genome is quite complicated, and with microarrays, as we'll see, they have limitations. But just to cover the basics, the human genome, it's roughly 3.2 billion, or milliarder, as we would say, base pairs long, and most of these letters, they don't vary at all. They're not variants, so to say. It's a loci or locuses. It's a location in the genome, but it doesn't vary because all humans have the same one insofar as we know. They're not polymorphic. And there are maybe 350 million variants. So a place is designated a variant if we've done some study and we've seen at least one person who seems to have a different letter at that place. And often these, of course, can just be a false reading when you're doing the genotyping. But it has been estimated that there are 350 that vary at least a little bit. But most of these are extremely rare. They may only be found in like one human on Earth, or 1 in 10 million or something like this. And so selecting on them would be useless since they don't really they're too rare to estimate their effect for, and they're not really contributing much variance. There's roughly 1 million or so common SNPs, and these are ones that have more than 1% frequency in humans, or common variants. And those are the ones we can deal with, and those are the ones that are causing most of the variation in polygenic scores. And there's a lot of easy stuff. Mostly when you're talking about SNPs, you're talking about these things: single nucleotide substitution. It's this one. Or polymorphism, this one's. And it's typically like a place where someone has a T and someone else has an A or something like this. And you can get things more complicated. You can have a short indel, which is, for instance, here we put in a C that some people have a C extra that other people don't have. Or in this case, some people have a T less. And these are the indels. The problem is you can also get weird stuff like insertions where like a big chunk is inserted here in the middle of, say, this green gene, or you can have like things can swap around. And so, for instance, an inversion, like things swap around like this one. So here the gene has been swapped. Normally it's read, say, from this side, but here for whatever reason has been inverted. There was a mistake. So you can really see that there's tons of different variations. And when you do GWASing, they're really only looking at these ones up here, the A's. These are the easy ones. There are a few studies with schizophrenia where they look at some of the B and C stuff, but mostly GWAS is concerned with the things up here. And this brings us to the problem is that microarrays, which is almost all GWAS data is based on microarrays. Microarrays is they're cheap, reliable, but they're limited because microarrays can only measure certain kinds of genetic variants. Microarray is essentially works the same way as a taste buds. There has to be a match on the one side and the other side, and it kind of reads the middle to see whether this one lights up or doesn't light up. And so, for instance, you make one here, you find something that has a unique pattern over here, a unique pattern here, and then you have something in between, and say, in here there can only be an A or an T, and so you make a probe that only lights up when you see an A and one that only lights up when you see a T, and you put genetic DNA into this, and you see if they light up. If only one of them lights up, it must be T. If both of them line up, it must be A and T, and so on. That's really how microarray works. It's an old but reliable technology, but it cannot really measure all these things like inversions and translocations and stuff. That's just fundamentally limited. And so we really need to move to sequencing so we can catch these more weird things like the rare variants and the inversions and all this stuff. Because we want to put all these kind of different variations into our genetic model so we can train and make it more predictive, because there's no guarantee that most genetic variation can be explained by these simple ones. Why would not a large proportion of it be explained by some of these? That's quite plausible. We just don't know yet because we don't really have good studies of these aside from a few schizophrenia ones. So we really need to do more sequencing, and we need this price to go down. So that's, as you can see, that happens already. So what we saw with the sequencing problem here is, or not, the editing problem is that genetic editing engineering requires us to know the causal variants. And since we don't know these, and we in fact cannot know these right now, since we don't have sequencing, we cannot really use much genetic engineering as it currently is, unless we get much better by some statistical improvement to identify them. And so it's not much used in animals either, because they have the same problem in animals. They just do microarrays and they predict stuff. They make a model that predicts, but they don't know why it predicts. They just know it works. We can get hypothetical here. We can say, what if we had a very reliable method of editing, and maybe some person, we know they have a bad version of this gene. We don't know why, but maybe we could just delete the entire gene they have, or we have this embryo with this bad version. We delete the entire version of gene ABC, and we replace it with, say, the version of ABC from Einstein, or some other, or maybe the average human, the modal genome, the most common version of this gene. So in that case, maybe we could make some progress with editing without having to know exactly the causal variants. But this needs a very large edit. Many genes are 10,000 base pairs long, so you need to write 10,000 base pairs without making errors, and without deleting the wrong genes, and so on. I don't think this idea has been well explored, but maybe someone will do it. Many people talk about CRISPR or Cas9 here, but in fact there's a bunch of other ones. Here's one review article that talks about, say, the ZFN method and the Talon method. And as you can pause the video and read the various limitations of this. There are a bunch of other ones of these that are currently being worked on, so maybe we will get to something that's reliable and can do large edits. So we can do this hypothetical idea here without having to know the causal ones. That would be interesting. I'm not really following this stuff much because it needs too much. It needs a lot of biochemical knowledge that I don't have, so I only watch from the sidelines. However, what we can conclude from this is that genetic engineering will not be good for some time. So maybe the next 20 years or so, embryo selection will dominate. Because you don't need to know causal variants for embryo selection. You just need to know something that's close enough in the genome that it predicts whatever you want. You just exploit the natural variation among siblings, which of course we know is large. And we can make models that predict between siblings, and we select the better embryo. And if you keep doing this, you will have to retrain the model on the newer ones. But this will still work. So things we can do here. We need to make the genetic models better. One way to do this is that we collect more people and we do these GWASs. And here I found this review that shows the improvement of the educational attainment IQ GWAS over time. And so they have names, EA1 and so on. So what we have here is that this is the Lee et al. From 2018, and they had 1.1 million people or so, and it could explain maybe 11-10% of the variation. And as a matter of fact, EA4 is now on the way. It has roughly 3 million people, and they find these 4,000 unrelated SNPs, and they also look at the chromosome X, and they try dominance. Unfortunately, they didn't find any dominant stuff. They can now explain 12 to 16, so we're gonna get up here, but we tripled this one over here, and we only got a little bit here. We can clearly see that there is a large diminishing returns in this approach to explaining. We're missing something. We need to have sequencing, or the model training is incorrect. There's some problem here, but still we're making some progress, and every variance point counts, right? They also do some sibling analysis, but unfortunately we don't have the paper yet. This is the abstract from a conference. So let's look at the more exotic stuff. Is that not only can we do embryo selection like we did, but we can actually skip generations. And so instead of doing embryo, we wait for the next person to grow up and then harvest their eggs. We can make stem cells, or we can take stem cells from the embryo, and we convert these to gametes. Gametes, remember, are the sex cells, so it's either semen or eggs. And then once you have these, of course, you can fertilize them across persons, or even within persons, as we'll see. And what you get then is you get more embryos, which you then wait, and you can keep this. You keep doing this. And so you start with some semen and some eggs. You do the IVF. You get hundreds of embryos. Then you grow these. You get the stem cells. That's what the ESC is here. And then you do the selection. You figure out which ones are good. You derive the new generation of gametes from them. You do the IVF again, and you have hundreds of embryos, but now it's the second generation. And you can keep going around this circle for a while until you find something that you think is good enough, so you're going to implement it. Or you can even clone it, or things like this. So that's where we need to go. In vitro breeding is what the animal breeders call it, and in humans it has been called iterated embryo selection, due to the paper by some rationalist who wrote this. And so this is where the good stuff is. This is where we need to go. As far as I can determine, this has not currently been used in animals. It's only hypothetical, and you can read this very nice review paper about in vitro breeding about it. I'm gonna guess it will be used within 5 years, so we need to make this 2 years. That would be very nice. So here are some of the papers that shows that this can be done not just in mice, but in cows, but also in mice. And I'm not aware of any paper that has shown this to be done in humans yet, but there are some papers that take some first steps in this direction. So of course, if we were serious about these things, we would massively state fund these things. We just need to get this going, right? Manhattan Project for embryo selection. So the next thing we have is that there's another method, in vitro maturation, and what you do here is that instead of putting in chemicals into the woman that fertilize some of these eggs and you get more of them out and you scrape them, the other option is that you put the syringe into the ovary and you extract a bunch of eggs that are not egg cells that are not quite ready yet, and then you get many thousands of cells and you mature them in the vitro, so in glass, and then you could potentially get hundreds of embryos or maybe thousands of them. And so this is maybe a sneaky middle approach. I've been reading about this for a few days, but essentially it seems to me that this approach is quite invasive because you have to do this extraction, and women don't like it, clinics don't like it, and so it's not really used much. It's still considered experimental despite having been around for 20 years. So maybe we already have a very good method, but we're not really using it properly, and maybe we can make this. The modern syringes are much smaller than the previous one. So I don't know. Maybe we can optimize this method to make it less uncomfortable, or maybe we just need to man up or woman up in this case and do it. So maybe we can get a lot of eggs easy this way, but I'm not sure. We can even get more bang for the buck, so to say, and it comes down to some clever stuff that has a physics analogy. And so what it happens is that when you make eggs, in this case, is that you start with one kind of pre-egg cell, and it divides internally. Then you do the swap, the recombination of genetics, and you end up with these two paths, which then divide again. The cool thing here is that you get these polar bodies. These are very small cells that they cannot really do anything. They just die, and it doesn't always divide, but these cells are useless. They just die off, and this one also dies off. However, because the egg cell here, the ovum, is based on the same DNA over here, which then split in two, this second polar body, it tells you the genetics of this egg, because you know the parental genetics, so you can sequence this one, and it will tell you by complement which ones are in this one. So that means that you can know the genetics of eggs without having to sequence them. And that's important, because normally when you sequence an egg, well, you destroy it and you can't use it. Of course, to read the DNA, you have to put it in a PCR and all this stuff. However, if you could use, for instance, this in vitro maturation and then track the second polar bodies, you could get the genetics of this egg. And then you have the genetics of the egg, and then in that case, if you could also do that for sperm, which has a similar process, you could avoid combining a good sperm with a bad egg, or vice versa. And this, of course, would increase the genetic variation that you could exploit. And if you could do this with sperm, of course, there's millions and millions and millions of sperm. And it is already possible to sequence these polar bodies, and you can see there are a bunch of papers on this. This paper is 2015, so this is not exactly new stuff. As I said, with sperm, you have the same thing, but instead of getting polar bodies, you get 4 sperm that work. Nevertheless, the same thing applies, that these sperm have complementary DNA, so you can sequence one of them and have the other one. However, to do this, you would have to do this entire sperm generation process in vitro. So you would essentially have to do something similar to women with in vitro maturation. You'd have to do some kind of sampling of men, or maybe you just want to do it from using the stem cell approach. I don't think this has currently been done. There are some studies that sequence single sperm. I have not seen any study that looked at this method for identifying genetics in the non-destroyed sperm, but that should be possible. Even more fancy methods are things like chromosome selection, and here you're really getting into the gritty stuff. You have your embryo, you sequence it, you know which things are on which genome, and so you see that, of course, you can make a partial polygenic score for any one chromosome, and you can see that maybe this embryo has a really good chromosome 5 and this other embryo has a really good chromosome 6. So wouldn't it be great if we could kind of put both of them in one embryo, create a kind of synthetic embryo? And of course, this would require you to figure out which copy of the chromosome things are on, the ones you want, because of course you have 2 copies and maybe only one of them is a good one, so you have to figure out which one is which, and you have to transplant it over without destroying it, and you also somehow have to sequence it. It's currently unknown how this would work, but if you could do it, you could boost things quite a bit more. Of course, the final frontier would be kind of back to the engineering approach, is that you could simply sequence or synthesize the entire chromosome you want. So you say, look, we've already figured out the good genetics on this chromosome. Why don't we just print a version that has the right ones? And this may seem futuristic, but in fact this has been done to some degree with bacteria that has very small genomes. And as I recall, we did synthesize some of these, and they work. So they do live. So we just need to scale up this process. Currently it's very expensive, but there are some startup companies working this area. So maybe if you're sitting out there and you're an investor, maybe you want to invest in some of these companies, see if we can push this technology around. That would or forward. That would be cool. Finally, this one is a lot of a boogeyman for many people. I wanted smart kids, but now they have green hair. That is not really much of a concern in real life. I think most of the problem I think will come from problems when you do editing. They will have off-target edits, right? You try to edit X, but you actually changed Y without knowing it. But in terms of not getting what you want for selection, it's not a big deal, because it's easy to calculate the polygenics or the genetic correlations between traits. And so you know that when you select for this trait by 1 standard deviation, you're going to get X standard deviation of this other trait, and it may be negative, so you get less of it, or maybe positive. And that's of course what all these genetic correlation plots tell you, is that, for instance, you select for intelligence, you're going to get less ADHD. The red color here is roughly minus 0.4. That's the genetic correlation. And so if you don't want ADHD and you want intelligence, well, you're in luck, because once you select for intelligence, you also select against ADHD, right? But also what you see here is you select for intelligence, you get a bit of autism. ASD is autism spectrum disorder. And so you already know what the trade-offs will be. And so any given embryo, you can calculate the polygenic scores, and you can kind of leave it up to the parents to say, what combination of traits is it that you want? Are you okay with taking slightly higher ADHD genetics in favor of, say, 2 standard increase in intelligence, and so on? And there's you can do this for every sort of thing, right? So this is really kind of a solved issue. I don't think we should be worrying much about this. As a matter of fact, of course, animal breeders who have been breeding cows for more milk and so on, they already know about the genetic correlation stuff, and they already deal with this by just looking at it and doing multi-trait improvement. You can read any number of papers. As you can see, a simple search finds like 7,000 papers. How far can we really go with this? There are many plots that go like this, but essentially we know already that we've been breeding chickens selectively for a long time. The chickens that grow fast and so on, those are the ones that current chickens are based on, and now we don't have to kill as many chickens to eat so much chicken meat. And we see the same thing with cows here. Actually, the cow population in US has been declining from a peak of 25 million or so, now down to 10. So the efficiency in per cow has actually caused us to need less cows or fewer cows. And so maybe we could accelerate this. This is only 2010, and as we saw, cow embryo selection started in 2008 or so with this bovine SNP. And so maybe we are up here now or something. I don't know. I didn't look it up. But so maybe we can get away with only 5 million cows now. I don't know exactly, but you get the idea. Humans, of course, the most interesting trait to boost is intelligence, because it seems to be the root of all good thing. Humans are mostly special from other species in that we're particularly smart, and we're not particularly strong or particularly nice vision or something, but we do have particularly big brains. We don't have direct intelligence measures of our ancestors, but we can track their brain size using the skulls. And if you do that, you get something like this, where current Homo sapiens is up here to 1500 or so. Of course, this is just the average, and some people are much bigger brains over here, and some are a bit small brains. So where we really need to go is, of course, here, right? We need to go infinite brain size. Actually, you don't want to go infinite brain size, because there's a limit to the function of a biological brain, but you get the idea. And then we can even get more exotic here, and using gametogenesis, that is, remember the place where you make sex cells, either eggs or semen cells from other cells, from stem cells. That also means, interestingly, that men can make eggs and women can make sperm. And so now you can see where this is going. So not only can homosexuals breed with each other and have kids that are descended from both of them, but you can actually fertilize yourself. Very interesting. There literally is a technical term for this. I swear it's real. It's called selfing. I guess some species do this already. The interesting thing about this is actually it will result in same-sex societies if you were to do it. And the reason for this is that women don't have any Y chromosomes. So all semen cells that women generate, remember the semen cell determines the sex of the baby. All semen cells from women will be X chromosome, and so all kids will be female. And so you essentially get this Wonder Woman society if this were to be done. However, in men, men can produce both, so they're going to get, when you make X based from men, you're going to get X with Y chromosomes. It's an abomination. It probably will not work. Nevertheless, the semen cells will have both. Men lack a womb, of course, so they're not really self-sufficient, but they at least can produce so gay couples can produce both, so to say. So what we really need is, of course, the final frontier. We need to have artificial wombs. It is the only solution to this trade-off with women, formal education, women career versus family stuff. If you had artificial wombs, they wouldn't have the pregnancy. And I mean, that is kind of a very transhumanistic future. And as you can see in sheep or whatever this is, a lamb, there is some progress being made on this. And so if we actually were to throw, say, $1 billion at this instead of useless educational programs, we probably could get there in maybe a decade or two. So it's a bit difficult to say, right? So, of course, many readers are now sitting there and going like, how can I get this technology now? And yes, you can get this technology at some companies. It's currently quite expensive because mainly the IVF is expensive. The egg extraction, all this stuff, you have to go to these IVF clinics. Everything medical in the US costs $1 billion. And so you want to do this in Czech Republic or something. But there are a bunch of rules, like only a married woman can do this. And I don't know what your family life is. But essentially, you can do this if you have enough money. And it may be worth it, depending on how much money you have. And if you have some like family disease of like breast cancer or something like this, you may want to do this because why wouldn't you? After all, if you can spend tons of money on private education for your kids that probably doesn't do anything, why not just pay up front for the genetic stuff that has a likely but small effect? It seems smarter. If you're like in the 20s, you may want to just store your eggs. Or you can gamble on these egg generation technologies coming of age before you turn too old. Maybe you want to save some eggs. You can always, as many countries, you can donate them away and they'll give you some money and you can save some of them. So maybe you just want to do this as a young woman. That would be sensible. If you can't wait, you're already like pushing 30. Well, I suggest you simply just begin with natural kits and you can maybe also freeze some eggs and then maybe later, when technology is improved, the next 5 years or so, you can do some selection on the next round. The important thing is to get started instead of waiting forever for a perfect solution. Remember the Nirvana fallacy. I, of course, have some recommendations. The only one I really have here is that you do not use this company because they're grossly incompetent. That is unfortunate, but that is the truth, and that is why I don't really talk about them. That's just because, well, they have issues. And so instead, what you want to do is there's a few other companies that are coming up in this. I think the one Orchid is currently the most plausible public one, but I also have knowledge of another one that's it's in the U-boat stage, it's hiding. And if you send me an email as a couple and you're interested in this, I will hook you up with the people behind this one. If you are more interested with reading, there's really two best places to go. It's you want to talk about all the technical stuff, all the details. I've been going over all these slides. You want to go to GWAS compilation and embryo selection. It really is the best resource on the internet for this sort of thing. It's quite often updated, as we can see was updated here in 2020, but probably will be updated soon enough. If you're more interested in the kind of philosophy or the ethics of this, then I recommend Creating Future People, The Ethics of Genetic Enhancement by Jonathan Anomaly. That's this guy. Based Jew. He tells us where things are going and why we want to go forward and why we shouldn't worry about the Holocaust and all these stuff. And so take it from a Jew that we shouldn't worry about the Holocaust. And with that said, of course, you know what to remember? More eggs. More eggs. All right. See you.